As promised, we're going to start this video with capillary types. We have three capillary types: the continuous capillary, the fenestrated capillary, and the sinusoidal capillary. So starting with continuous capillaries, these guys are formed by endothelial cells. So we have an endothelial cell here, we have a basement membrane surrounding, and then we have, here's the nucleus of the endothelial cell, the lumen is the opening in the middle, and then those are red blood cells. We have intercellular clefts and gaps in continuous capillaries. Where we find these guys, skeletal and smooth muscle, we'll see continuous capillaries to the connective tissues and in the lungs. Fenestrated capillaries, these guys have a plasma membrane, and that plasma membrane has a lot of holes in it, or fenestrations. A fenestration is an opening, a hole, a pore, whatever term you use to picture in your mind that I have some openings within that plasma membrane. We're going to find these guys in the small intestine. We're going to find them in the kidneys, the choroid plexuses. Remember in 201, the choroid plexuses were lining, or were those vasculature regions in the ventricles in the brain where we produce cerebrospinal fluid. We'll see fenestrated capillaries at the endocrine glands. And again, we're using those fenestrations as exit points. We're going to allow things to exit or enter through those in the capillary. Our third type that we're going to talk about are called sinusoids. Sinusoids are another form of a fenestrated capillary, but in this case the fenestrations are very large. They've come together, they've coalesced, and we've got these much larger openings. They look much more like Swiss cheese to me. The other thing that we'll notice here in the sinusoidal capillaries is that we have an incomplete basement membrane, whereas in the continuous and the fenestrated capillary we had an intact and completed basement membrane. Here in the sinusoids we have an incomplete basement membrane. It may also have some holes or some fenestrations in it. We're going to see these types of capillaries, these sinusoidal capillaries, in very high tissues with a lot of blood in them, that typically hold on to a lot of blood and typically do some important cellular exchanges. So liver, bone marrow, spleen, the anterior pituitary where we've got a lot of hormones moving, parathyroid gland, those sorts of places. So those are your three capillary types: continuous, fenestrated, sinusoidal. You want to know how they're formed, what they look like, and where they're located. The whole point of a capillary is to have exchange. So we have three forms of exchange. You can see two on the slide right at the moment, and we'll add the third in just a second. But the most important method of capillary exchange is called diffusion. Diffusion is something that should not be new to you. It is a passive process. When we talk about capillaries, we'll talk about diffusion of oxygen and carbon dioxide. Those two items will diffuse across capillary membranes easily. Glucose, amino acid, all of the plasma solutes except the large proteins are all going to pass very freely across these capillary endothelial cells. They're going to pass through the lipid bilayer, they're going to pass through the fenestrations or the intercellular clefts. The exception to that is the blood-brain barrier. So on your slide you'll see BBB. BBB stands for blood-brain barrier. It does not allow for the diffusion of water-soluble substances. It is non-fenestrated, so there are no openings in it. It's a non-fenestrated epithelium. It has tight junctions, and the reason it is formed that way is it's a protective measure. It's to keep pathogens from the blood from entering into the brain, and that that major control center. The downside of that is that it also really is difficult for us to pass medications through the bloodstream to the brain. As promised, the second version of capillary exchange is called transcytosis. Transcytosis allows for the passage of substances across the endothelium in tiny vesicles. We're going to utilize endocytosis and exocytosis when we move across the endothelial membrane, and so we're going to use this method for large lipid insoluble molecules, things that can't pass that phospholipid bilayer easily, things like insulin or maternal antibodies that are going to pass through the placenta into the fetal circulation. The third version of capillary exchange, which you are responsible for, and we'll come back to it, so I want to touch on some other things before we do. In fact, I want to go through the venous side before we get to bulk flow. So what you do need to know is that we're moving substances in and out of the capillaries. We're doing it through three processes, either diffusion, which is the most important method, transcytosis, which is going to work with those large lipid insoluble molecules, and we're going to use vesicles through endocytosis and exocytosis, and then bulk flow. Bulk flow being the last one, and we'll pick that one up again in a little bit. Our next video will start tackling the venous side of circulation, and we'll move on from there.